So we work on certain quality control chaperones as well as proteases. We are interested in the basic biology, but also we're interested in targeting these quality control machineries to develop new antibiotics or anti-cancers. So proteins are basically the workhorses of the cell. They make the cell what it is, they define the cell as well as allow the cell to divide or migrate if need be. And therefore, in the cell, there's these quality control machineries that ensure that a protein is properly assembled, is properly folded, and also is properly integrated with other machineries. Also, there are proteases. So they ensure that when a protein is not behaving well or it's time to get rid of it, that protein is removed from the cell. What we are trying to do is trying to identify compounds that affect the quality control mechanisms in the cell, and we are hoping this would lead to cell death. One way to think about it, basically, let's say you have a small puppy chewing your sofa, your carpet. So basically it's now just hyper and eating everything up. This is one way we are targeting cancer cells. We want to get rid of those cells. And this is one way these new compounds that we describe in that paper work. So by dysregulation. And this is a very different mechanism than what is typically done. Many compounds actually inhibit a protein, prevent it from doing something. But these compounds actually hyperactivate. They cause the protease to just go wild. The C.l.s. is one of the facilities that we really like. The people there are quite knowledgeable. The synchrotron technology is actually extremely important for us. We are doing what we call basically structure-based drug design. So we need the structures to be able to see how the compound is binding and to further optimize that compound. So we are hoping that these compounds would eventually get into the clinic to target certain cancers.